Hello. 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 Guys hear me? Sweet. You can hear me. Excellent. What's up, Dixons, Carrie and Z, HB Wilder, Leo Dallier, Mr. Metric, KJ. Hello, everyone. Glad to have you guys here. Mr. Metric has a hydroponic tank set up outside, ready to grow some not yet GMO plants. What are you guys growing? What are you growing in your hydroponics? I'm interested in hydroponics. That sounds really cool, actually. How's everyone's weeks going? It's Thursday. It's the end of Thursday. I'm super excited about Friday. Friday. Quick question from Dixon's. The LB amp media you sent us had to heat in the bottle. So there's LB amp agarose media, which goes on your plates. So that needs to be heated up. But there's LB amp broth, which is just a liquid. It just needs to be dissolved. So no heating needed. You just shake it up real quick. So you add the bacteria. Yeah, so like in the broth, you don't actually heat that one up. But if you do end up heating it up, it's okay. You could just let it cool down and then inoculate the bacteria. Oh, hello. Mr. Metric's growing some peppers, dandelions, hawkweed, carrots, grapes. It's pretty cool. Spearmint cutting. You're so nifty, you guys. Yeah, Dixon's. The broth doesn't need heat. Just put it in the water, shake it very vigorously. Doesn't matter how many bubbles you make, and once it all dissolves, it will turn clear. Not meaning the yellow will go away. It won't turn a different color. Once it turns clear and no floaty particles, then you can use it. Inoculate your bacteria in it. Okay, well, I'm gonna get started. It is 5:02, and yeah, if you have any questions, please feel free to ask on the chat, and I'd hope to answer them all. Yes, Dixon's, no microwave. No microwave needed for the LB amp broth. No microwave. Okay, so let's get started. So I don't know if how many of you have read the really cool paper called "What Does It Mean to Be 75% Pumpkin?" That is in the 4th week, and it goes over what we learned last week, which was transcription, translation of DNA, the central dogma of biology. So, yeah, if you've read that, cool, you're all caught up. But if you haven't, I'm not going to spoil it for you. It's a really, really great paper. I highly recommend you go read it. It's a short paper, so it's not like one of those like, things that you just like read like every line of and try and retain that information. No, it's kind of a very conceptual thing. I'm gonna go over that real quick, just a little bit, and then not give away the ending. No spoilers. And then we're gonna go over some antibiotic calculations that you will be needing in the very near future for, I believe, this week's experiments with kanamycin. The PDUSC-PDON experiments that you will be doing in the next coming weeks. So I will explain to you antibiotics. Ted Reynolds asks about the previous experiment, the glowing bacteria experiment that we just completed this week. And the glow usually lasts between anywhere from 24 to sometimes 48 hours. You can actually keep the glow going as long as you replenish it with media. So you can do that by making a new batch of media and taking just like a small tiny sample of the glowing bacteria and inoculating the new one, so you could just keep it consistently glowing. I've seen people make really cool glow lanterns out of it. Um, I've seen someone make on Halloween, like carved out a pumpkin and then put the glowing bacteria and covered the pumpkin's insides to like glow. Lots of really cool applications that you can do with the glowing bacteria. So as long as you feed it, it'll keep glowing, which is cool. Okay, so let's go over the what does it mean to be 75% pumpkin. Um, Want to emphasize there's no such thing as a model organism. Model organisms are bullshit, as you found out in our syllabus, because there's no such thing. There's no such thing. When people are like, oh, that's not a model organism. It's not true. It's just not very well known or sequenced yet, and that's fine. That's fine. So in the paper, it tells you some claims about science and some pitfalls that the claims lead to. So a lot of people, when you talk about comparative genomics, people are just like, oh, it's obviously like how closely related you are to like some organism, and it's somewhat true, but not really true. So comparatively, we can't really compare ourselves to a pumpkin. Um, so in the paper, it kind of goes about and describes what we learned last week of the central dogma of biology, how on the raw form of DNA, your A's, T's, C's, and G's, and all the way to proteins don't really translate very well. Remember that when we do transcription translation, DNA goes from RNA to protein, and within the process of DNA translating to RNA, um, things are translated and then transcribed into transcription, transcribed, translated. Yeah, translated into protein, which means that some of the DNA may be excised out from our introns and exons. So not 100% of DNA becomes protein. Now, with that said, a lot of people claim that DNA is like, the code of life, which it is. But we kind of have to step back and look at the organism as a whole. Because remember, not everything in DNA translates directly protein. And once it becomes protein, it may not express in humans completely. Lots of proteins make up hair. Lots of proteins make up teeth, eyeballs, saliva, anything. So it's really difficult to say that this specific sequence of DNA will completely be like, an eyeball or something. So a lot of people in media obscure things like, oh, we're very, very closely related to, like a plant because of this, because of X, Y, and Z. Well, no shit, Sherlock. Like, all the functions of a plant can function or have function in a human. So the best way to understand genomics and how it's progressed and evolved throughout history is by looking at our ancestral origins, right? So, for example, when people do a lot of mammalian or human cell culturing or things like that, most of the people look at the mitochondria. Mitochondria, what is it? I'm sure you guys learned in biology or science 101 in your grade school on the SAT. Mitochondria is the powerhouse. Mitochondria is a, wait, hold on. It's mitochondria is the powerhouse of the cell, right? Oh my God, I totally botched that up. I hope I edit that in post-production. Mitochondria is the powerhouse of the cell. Say it loud, say it proud, because that's the only thing that you should have learned in grade school. So what that means is mitochondria is very, very well conserved throughout all species because lots of things have mitochondria. Plants have, or I'm sorry, lots of humans, lots of animals have mitochondria. So if you go into the mitochondrial DNA, super well conserved between males and females. So when you procreate and have a baby, you're going to get the mitochondrial DNA from your mom or father, which is pretty cool. So you can actually trace the lineage back and understand genomics that way. So throughout time, like that is that portion has been very well conserved, and yeah, you can compare genomics with that. So now let's go back into functional genomics. So functional genomics, something like if we compare chimpanzees to humans, we have evolved a opposable thumb. And that indicates that we have something that chimpanzees don't. And it's been well conserved throughout the human species. Now it's kind of hard because you can totally sequence a chimpanzee and you can sequence a human, and I believe the percent that is different in our DNA, in our coding DNA, is only like 2%. And so people are like, "Oh my God, we're super related to chimpanzees. Like they're going to be our overlord soon." But maybe not. I don't think so. Like the things that translate into protein, opposable thumbs, were far, far more evolved than chimpanzees, even though we are 2% off, right? So the paper kind of goes through like debunking why it is incorrect to say we are 75% pumpkin, because we are in the sense of DNA, but when we look at comparative genomics, we are nothing like a pumpkin. Yeah. Okay, so let's talk about calculations. So go ahead and read that paper. It's really, really interesting. You can find a little bit more about genomics and comparative genomics and how things have been evolved. And it'll really hone in on those skills of transcription, translation, and the central dogma of biology, and also the mitochondria as a powerhouse itself. Yeah. All right. So let's dive into it real quick. I'm growing smaller. I'm growing smaller. Okay. How to make media. So, we kind of cheated because the first couple of experiments, you all got everything nicely prepackaged. Everything was in a tube with really nice instructions, and it was so great. Life is so great. But as a biohacker, you're not going to have everything nice and packaged for you unless you obviously order directly from us, which we totally will package that for you. But everything is not super nice and packaged. Usually biohackers that have labs in their homes will purchase in bulk, will make media from scratch by themselves. And it might be challenging to kind of like go about that and see like what needs to be done or what things you need to like start your lab, for example. But there's lots of resources online, and And I can send you guys a few links to see like how would you go about building your own lab in your own home to become like a really cool biohacker. But the first things first, we need to know how to make media from scratch. And that is a skill that I really want to teach you guys all. So this is how to make media. All right. I'm going to kind of quickly go over the calculations. Bring out your calculators. This is a very two-way conversation. I want you guys to interact with me on lines. And yeah, so let me know some of your answers as we go through it. Okay, you guys ready? Get your calculators. Yeah, you can.
Will we get a ticket?
No.
We get to Why?
Because it's property. It's our private property. Hurry up. Like, yep. Okay. How to make medias. Medias. Medino. Medias. I can't see you. I make pancakes. But you can see me, right? I don't know what you're doing, or I don't, like, yeah. It'd be kind of creepy if I could see you all. Anyways, so how to make medias. Oopsie. There are two different types of media. Oh, I'm squee. My bad. All right. So there are different types of media. There's culture media, there's minimal media, selective media, differential media, transport media, and that's it. There are lots of types of medias. You don't need to know all of them. All you need to do is know a few of them. So I'm going to quickly go over them myself. Culture media means just like regular broth, it's enriched with all the essentials such as sugar, water, vitamins, essential needs to essentially make you alive. So that's culture media. It's not being cultured or anything. It's just the bare necessities to live with enrichment. Minimal media is just like even barer than that. So minus all the vitamins, it's literally just like sugar water. Very, very minimal media. Selective media means like something that you supplement with like either like with like antibiotics, something that you can distinguish between a negative finding or a positive finding. So for example, our PDUSC PDON plasmids that we sent you guys, it grows in kanamycin. So if you do not add the kanamycin, anything and everything will grow. However, if you add the kanamycin antibiotic into the media, then it becomes selective. And then that way you can tell, yes, this is the correct type of bacteria that I'm trying to grow. So that's what selective media is. A majority of experiments are always saying like, okay, does it grow? Does it not grow? And then kind of expand your experiments on that. So selective media is actually very, very well used within industry and practices. There's differential media and transport media. We won't really talk about these, but they are used in certain situations for different types of experience. I had to say goodbye to my friends. And then there is the liquid versus agar. So liquid is the broth that you don't need to microwave in. You literally put it inside of water, you shake it up really well, and then once it dissolves, then you can inoculate your bacteria. Now the other is agar, which is something that you need to microwave and pour plates and then streak it out on an agarose medium. A good way to subculture some of your stabs that you've already received is by making an agar plate And then whatever the agar plate is made of, let's say for like the LB, or I'm sorry, for the P dusk, P dawn, you can put it on kanamycin LB agarose plates. So then that means you need to modify your plates to provide kanamycin versus like the liquid. And so once you streak it out on the LB kanamycin agarose plate, then you can actually take big, huge globs after you've grown it up overnight. Take big, huge globs, inoculate it into the tube of liquid agar, or of liquid broth, my bad. So people that are starting to do the GFP bacteria glow experiment, if you want to get even more glow, you need to put in more cells. So a way to achieve this is first put it on the agar plate, the LB ampicillin agar plate, grow it up. You're going to have big fat colonies in the next couple of days. Take a huge loop of it, literally just like scrape it off and then just try and inoculate it. You're going to get a really, really nice bright glow. Just make sure you have a lot of food for it, or otherwise it's going to grow too fast and it's going to just expire out the media and you'll never see glowing. So that's a really cool way to differentiate. All right, back to the calculations. So usually plates need 1% of LB agar. It depends on the organism. If I've seen it go up to like 10%, like 2%, but 1% is a really good baseline for things like E. coli, because you need it to just like the wavelengths to penetrate the agarose. It can't be like too thick or too viscous. So a 1% weight per volume LB agar is generally used in labs. So before you start, like weighing out stuff on your really cool scale, um, you count how many plates you need per experiment. So you're like, okay, well, I want to make an experiment. I want to make one plate that has my experiment on it, but I also want to make like, a negative plate, or like expand my experiment out to like 4 plates and see what it does on 4 plates. So you think about like how many plates you want to make. And each plate is 30 to 40 milliliters per plate. Now, it's totally a ballpark. I don't like necessarily measure every single like tube out 30 or 40 milliliters, but generally, if you're pouring a plate halfway on the plate marker, it's usually generally 30 to 40 milliliters per plate. But make sure you just make a little bit more just in case you overshoot on the plates. So maybe add like, I don't know, a couple, like 20 more microliters to the reaction, just in case you lose some, right? Or boils over, God forbid. So then you figure out, okay, how much is the total volume you need? Benedict Noel asks, why do you need the microwave for the agar and not the liquid? So I don't know if you've ever made jello, but the agarose is essentially gelatin. You can actually eat the agar. I've done that before in a lab. I would not recommend trying that because it totally violated some OSHA laws. But I was dared to eat agarose, and it's literally jello. So if you've ever made jello, you have to heat up the gelatin, right? And that's the agar part. Now the LB part is the broth, is the enriched media. So it mixes with the agarose, and you have to microwave the whole thing and then cool it down and then pour it in a plate, and then it'll solidify really nicely in the plate. So you get this really nice rich media jelly plate. And you could probably eat it. It will not harm you, without the bacteria, of course. So that's why you need a microwave for this stuff. Jello. All right. So where to find Agar? You can find it on our website at theodin.com and lb-agar. Yes, you can find more. If you want to make more plates. All right, we're going to go to the antibiotic selection. So we provided you an antibiotic called kanamycin. So you can tell if a chemical is an antibiotic by the ending word mycin. And generally, antibiotics need to be stored in the fridge. So today, I'm going to teach you how to make a working stock, and then also to make your agarose plates from the working stock, because right now the concentration of kanamycin in that tube in powder form is like 99.9%. So it's essentially 100%. And what you need to do is dilute it down in your working stock. And then once you dilute it down, you need to even dilute it even more into your agarose to get a 1% agarose gel with kanamycin, with 1x kanamycin. So on your plate or on the tube, it says 30 milligrams per milliliter, which is 51.50 micromolar. Oh my God, maths. I know, I'm freaking out too. It's okay. It's fine. I'm here to teach you maths. I'm here to hold your hand in this, because maths can be really hard, but we're all going to be fine. All right. So, let's make a 100 microgram in a milliliter in 150 milliliters of total solution. Okay, so then that means I said, okay, like I want to make 150 milliliters of total solution, and I need to make this type of concentration. So then let's do an example. Ampicillin. I'm not going to give you the answer for kanamycin. That's something for you to figure out. So we're going to do an example with ampicillin. Ampicillin is 100 milligrams per milliliter. You're going to do exactly what Alexis Ortega said, is the M1, or the V total equals N1 over V1 equals, I'm sorry, my bad, N1 times N2, blah. Oh my God. So it's the total volume equals M1 V1 times M2 V2. I don't know why that was so difficult for me. It's Thursday. So then you say, all right, I have 100 milligrams over 1 milliliter, and I want to figure out how many milligrams for the total of 10 milliliters of the total solution. And then you're going to convert from milligrams to grams, which is 1,000 milligrams per 1 gram. So then you get 1 gram of ampicillin in 10 mLs of water. Because in the little tubes that you get, I think they're like the 15 mil Falcon tubes that you just get arbitrarily, it'll make you 100 milligrams per milliliter of ampicillin stock solution concentration. I generally keep it at 100 milligrams per milliliter. It's just way easier to work with, because it's intense. Like 100 is very easily dividable. So I usually generally make 100 milligrams per milliliter for working stock solution. So once you calculate all this, and then you make it, then you should store it inside of your refrigerator at 4 degrees Celsius, because antibiotics degrade over time, and it could just last longer in your fridge. Now, if you leave it out, it might compromise the antibiotic, and if you put it on your plate, sometimes the bacteria will not grow. So always keep a fresh stock solution in your fridge, always. Make sure when you use it, you put it back in the fridge, because it's temperature sensitive. So yeah, here's what I pretty much botched. So yeah, M1 V1 equals M2 V2. This is a very simple calculation, and we will totally be working with it. So we're going to plug our total volume, remember, at 150 milliliters, right? And we're going to do some maths. We're going to try and obtain 100 micrograms, or I'm sorry, 100 milligrams for the 150 milliliter. And we're trying to get a concentration again of 100 micrograms times V2, which is what we're trying to figure out. So then we need to figure out how to convert micrograms into milligrams, right? So this is the conversion right here. And you're just going to plug that back in. And you're going to get a V2. Can you see the last part of it? Probably not. Oopsie. Ah, yes. And then you're going to convert the answer of 0.15 milliliters back into, oh my God, I'm sorry. No. Into microliters, right? So that's 1 milliliter equals 1,000 microliters. What does that mean? All right, you need to get 150 microliters to your agarose solution. Now you have 100 micrograms in a milliliter of 150 milliliters in total solution. Follow? All right. Try it out with your kanamycin. Confirm your answers with classmates or instructors. Just kind of practice your maths. I know it's hard. You could totally follow along from this tutorial, or you can kind of finagle with it. I've posted this lecture inside of the Google Classroom, so make sure you take a look at it. If you still have questions doing maths, don't worry about it. Just email me personally, and I'll take you step by step through it, and then try and make you understand, why we do this kind of thing. Because, yeah, hand holding in the biohacking community is not very prominent. So yeah, I'd love to help you guys out with some of your calculations so you could do this readily in the future. All right. There is some debate about M is milli, but I said micro. Okay, so M, just the regular M, is milli. And then the U, it kind of looks like a little U, UL, is micro. I'm sorry if I botched it up in explaining things. It's difficult to explain things while you're kind of like on the fly, especially with such small amounts of M's and U's. But it's all in the document, so it's very, very clear out there. Does a solid kanamycin need to be refrigerated? We do refrigerate ours only because we have like a kilo of it. We just don't want it to like degrade. You can do the same thing, but in powder form, it's pretty stable. Once it gets diluted down into liquid form, it is a little less stable. So you don't need to. You can. It's fine. Any other questions, comments, concerns? I know I kind of blew over that really, really quickly. Um, it's for me, I'm trying to explain it very like rationally. This is in PowerPoint form. It's hard to lecture in PowerPoint form without sounding kind of robotic. But, if this video didn't make any sense, I highly recommend you just kind of like struggle through with it on the Google Classroom. And then if you're really, really, really confused, contact me or contact some of your classmates that understand it better. This is a really important concept, and I think that everybody should be learning it. Yeah, Constantine. So, you know, I did it really fast. Just go back to Google document and then just like struggle with it because that's the best way to learn really quickly. If you're really, 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 really lost afterwards, contact somebody from the class or me. Yes, Constantine theory, you are diluting it twice because remember the first, in powder form, it's 100%, and we want to get down from like 100% to like 100 micrograms, right? Which is like really, really diluted. So essentially, yes, we are mixing it with water, the kanamycin, diluting it down from powder form to liquid form by a lot. And then from that, we're going to even dilute it more down and put that into agarose. So that's essentially the workflow. But once you pretty much like get from the regular stock to the working stock, you can keep that for a while so you can make other plates with it. Yeah. So if you have any questions, conceptual questions, feel free to ask now. It's hard working with like really tiny like micro amounts. I don't think there's any other profession that works in like micro amounts. We work in like micrograms and microliters. We're working with tiny things, so it's only right that we use micro. All right. Without any ado, if there's no more questions, any questions? Doo-doo-doo-doo-doo. Any questions? I will end the stream. What is next week? What are we working with next week? Week 5. Oh my God, we're like halfway through the class. How insane. So next week there is live stream same time, an experiment, the bacterial color kit. Are you guys excited? And there's actually tons of papers. We're also going to be going over some bioinformatics. So make sure you download, I believe it is, oh my God, what is it? Make sure you download SnapGene Viewer. So there's SnapGene, SnapGene, SnapGene. There's regular SnapGene and there's SnapGene Viewer. And SnapGene is the paid version, and then SnapGene Viewer is the completely free version. So we want to keep this as low cost as possible. So SnapGene Viewer is sufficient enough. So make sure that you download that prior to the class so you can follow along with it. We gave you a few sequences and sources in the Google Classroom for you to play around and understand and see some of the raw data that we've provided for some of the GFP stuff, which is pretty cool. Yeah. HB Wilder says, a couple of agar plates in my fridge have white stuff growing on it, but others don't. Do you think they're all contaminated? Just revealed itself yet? Just not revealed itself yet? Definitely throw away the ones with the white stuff on it. Any growth so far on those plates, they're definitely compromised. You don't want it to spread to your other plates. So throw those away first, and then keep the plates that you think that don't have it, and just kind of like see if it's contaminating it more. If it's contaminating the rest, then just throw away the rest. You don't want to ruin your experiments based on just like, oh, there's like a little white patch, and then like several weeks of experiments have gone down the drain. Yeah, it's okay. I mean, like, once you make a batch of plates and like one gets contaminated, it can be from anything from like you accidentally opening up that one plate or something. It may not necessarily be for the rest of the plates, so it's fine. Alexis Ortega, any references on getting all the things for labs, including antibiotics, etc.? I can definitely give you a link. There is a great source that one of our biohackers in our community actually made this really nice comprehensive like how to get a lab in your house. And it goes over like where to source some things. Some of like the lab equipment I actually purchase on eBay. So for example, like a PCR machine, I can get it on eBay pretty cheaply. Or if I'm trying to find an incubator, I kind of troll Craigslist. Or if you have a science or like a university around you, sometimes they have like sales of like trying to get rid of inventory. And sometimes you can poach some of their sales, go to some of the auctions. Um, yeah, those are a few ways to source things. Things like media, such as like LB broth in bulk or like antibiotics, buy from China. They usually ship in bulk, so if you're looking for like 50 kilos of LB broth like for pretty cheap, I would get it from China. That's where we get our stuff from. It's worked out really well because we have so many students and you guys all need the media. So, but for like a individual biohacker, it's hard to just like drop 10 G's on like several kilos of like kanamycin. We do sell antibiotics in really small quantities in our store. So, yeah, plenty of resources to get stuff. If you haven't joined the biohacking and genetics group on Facebook, I think it has like about like 3,000 people that follow it. It's just a bunch of biohackers. Everybody knows really well how to source a bunch of these things. So if you ask like, hey, where can I find like this antibiotic? Or hey, where can I find this media? People will be like, oh, here it is. And here's like how cheap it is. Or like, hey, I'm trying to find like a cell line. Like, can anybody provide me with it? Usually with like a small shipping fee, they'll like ship it over to you because people are nice like that. Yeah, Daniel Mallet really, really copped out with this CO2 incubator. He got the CO2 incubator that probably is like an anaerobic chamber for like $10 at a university auction. So, some good shit, man. Yep, and Mr. Metric says government surplus is useful as well. You could definitely get like a really nice refrigerator or anything from that. I will provide you with the link, but without any further ado, I will say goodbye to you. Goodbye.